Deoxyribonucleic acid is the molecule we have inside our nucleus that holds all of our genetic information. There are three parts to a nucleotide. There is a phosphate group, a sugar, and then a nitrogenous base. The four nitrogenous bases found in DNA are adenine, guanine, thymine, and cytosine. Adenine and guanine are both purines. Thymine and cytosine are pyrimidines. DNA wants to have a purine-pyrimidine pair. So A binds to T and C binds to G. In A and T, they need 2 hydrogen bonds, and in C and G, they need 3 hydrogen bonds. When these two sequences of amino acids come together, we end up with a double-stranded molecule of DNA. So in our DNA, we have deoxyribose is the sugar. We have 4 nitrogenous bases. We have A, G, C, and T, and they form a double-stranded double helix. In RNA, RNA has a ribose sugar instead of a deoxyribose. It has 4 nitrogenous bases. It has A, G, C, just like DNA, but instead of a T, it has a U for uracil. And generally, RNA is single-stranded. So we can take the information from our DNA, turn it into mRNA, and send that out to the cytoplasm in order to make a protein. So when we take our DNA and we pair it with our RNA, A is going to bind to U instead of a T. C is going to bind to G, G is going to bind to C, and T is going to bind to A. So our big substitution is going to be when we have an A in DNA, we're going to have a U in RNA instead of a T. When we go from RNA to RNA, A is going to bind to U, and if we have a U, it's going to bind to an A. There are 3 types of RNA. There is messenger RNA, where we use it to send the message from the DNA out to the cytoplasm. We have transfer RNA, and these are the molecules that have an anticodon on one end and an amino acid on the other. And then we have rRNA, and that combines with proteins to form the ribosome, where translation is going to occur.